Welcome you all. I am Leema Rosemary, Assistant Professor, Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. Now I am going to talk about biotransformation. The term detoxification or detoxification refers to the series of biochemical reactions occurring in the body to convert the foreign compounds to non-toxic or less toxic and more easily excretable forms. In recent years, the term detoxification is replaced by biotransformation or metabolism of xenobiotics. Man is continuously exposed to several foreign compounds such as drug, pollutant, etc. These foreign compounds produced in the body are toxic effects and therefore they should be quickly eliminated from the body. This is known as detoxification. Site of detoxification. Detoxification reactions are carried out mainly in the liver, which is equipped with the enzyme machinery. Kidney and other organs may sometimes be involved. All xenobiotics taken up from the intestine are transported to the liver by a single blood vessels. Metabolites of xenobiotics can be excreted by the kidneys or via the bile. They can also be exhaled via the lungs. What are the steps involved in biotransformation? It can be divided into two phases, phase 1 reactions and phase 2 reaction. Phase 1 reactions involve oxidation, reductions or hydrolysis. Then phase 2 reactions involve conjugation. Phase 1 reaction, oxidation, reduction, hydrolysis. It produced metabolites, then it converted to soluble form and finally excreted. And xenobiotic compounds also involved in phase 2 reaction, that is conjugation reactions. Here the product combined with some compounds, glucuronic acid, glycine, etc. Then it converted to soluble, finally excreted in kidney. This is the flow chart for the biotransformation. And the toxicants stored in the fatty tissues. So the compound lipophilic in nature, then it converted into hydrophilic and finally eliminated from the body. Phase 1 reactions. This reaction is the first step in xenobiotic metabolism. The majority of metabolites are generated by a common hydroxylating enzyme system known as cytochrome P450. It's a group of enzymes that professionally oxidize xenobiotics in phase 1 reaction. Oxidation. A large number of foreign substances are detoxified by oxidation. These include alcohol, aldehyde, amines, aromatic hydrocarbons, sulfur compounds, etc. First of all, alcohol, aliphatic and aromatic alcohol undergo oxidation to form the corresponding acids. Example, methanol, it converted to formic acid. Then next, ethanol, it converted to acetic acid. Then benzyl alcohol, it converted to benzoic acid. Then aldehyde are oxidized to produce the corresponding acid. Benzaldehyde to benzoic acid, chloral to trichloroacetic acid. Then third, amines and their derivatives are also oxidized. It converted to the corresponding acids. Example, aliphatic amine, it converted to aliphatic acid and urea. Then aniline converted to para-aminophenol. Aromatic hydrocarbon also oxidized to mono, di, trihydroxyphenols. Sulfur compound oxidized to sulfuric acid. Some drug converted to soluble form, that is non-toxic nature, then excreted in urine. Here the example, meprobamate, it's a tranquilizer. It oxidized to hydroxyl mepromate and also excreted in urine. Then reduction reactions. A few examples for the reduction reactions are picric acid, chloral, nitrobenzene. Picric acid converted to picramic acid, chloral converted to trichloroethane, and nitrobenzene also converted to aminobenzene. Then hydrolysis. Several compounds undergo hydrolysis during the course of their detoxification. The hydrolysis of the bonds such as ester, glycosides and amide. These include aspirin, diisopropyl fluorophosphate, atropine, procaine. Aspirin is a painkiller tablet. It hydrolyzed into salicylic acid and acetic acid. The next example, diisopropyl fluorophosphate, it converted to dialkyl phosphate. Atropine converted to trophic acid and tropine. Procaine converted to para-aminobenzoic acid and diethyl aminoethanol. Then phase 2 reactions. Phase 2 reactions refers to the second step in biotransformation. It usually means that the oxidized compound is conjugated with an endogenous molecule. This reaction increases the water solubility. Further, many conjugated metabolites are actively excreted via the kidneys. These reactions involve the covalent attachment of small hydrophilic endogenous molecules such as glucuronic acid, sulfate, glycine to form water-soluble compounds that are more hydrophilic. This is also known as a conjugation reaction. Several xenobiotic compounds undergo detoxification by conjugation to produce a less toxic compound and excreted easily. Conjugation, here the enzymes involved in this step, transferases. This is a group of enzymes. It conjugate xenobiotics with endogenous compound. In the process of conjugation may occur either directly or after phase 1 reactions. 8 different conjugating agents have been identified in the body. There are glucuronic acid, glycine, cysteine, glutamine, methyl group, sulfate, acetic acid and thiosulfate. First, glucuronic acid is the conjugating agent. It's mainly involved in the conjugation process. The active form of glucuronic acid is UDP glucuronic acid produced by the uronic acid pathway. The microsomal enzyme UDP glucuronide transferases participate in the glucuronide formation. This enzyme induced by some drug, example barbiturates. Glucuronic acid conjugation may occur with compounds containing hydroxyl group, carbonyl group, sulfidyl group or amino group. Here the example for conjugation, benzoic acid conjugate with UDP glucuronic acid and then it converted to benzyl glucuronide plus UDP. The next example, bilirubin, it's an important one. Daily form actually this is the waste excretory product produced from body. Daily the bilirubin excreted through the urine and fecal matter. So bilirubin is an insoluble compound. When it combined with UDP glucuronic acid, then it converted to a soluble one that form bilirubin diglucuronide. Next example, glycine. Glycine also a conjugating agent. It combined with many compound. For example, benzyl CoA. It combined with glycine to form hippuric acid. So hippuric acid is excreted compound in cows and horses urine. Then another example, phenylacetic acid react with glycine to convert it to phenylacetic uric acid. Then cholic acid plus glycine converted to glycocholic acid. Then third conjugating agent, glutathione. It is an endogenous molecule. It's a tripeptide. It conjugated with xenobiotics in phase 2 reactions. Glutathione is depleted toxic reactions between activated xenobiotic metabolites and proteins. Lipids or DNA may occur. Next conjugating agent, thiosulfate. The highly toxic cyanides are conjugate with thiosulfate to form less toxic thiocyanate. Thank you.